Hello guys, as promised on Thursday, here is a video about enzymes which is going to complete this chapter. We said that enzymes are biological catalysts, which means that they do help chemical reactions to occur. Now, as you may recall, most chemical reactions that occur in our system are intended to occur. Either those chemical reactions are spontaneous or non-spontaneous, they are intended to occur. However, the limit of occurrence of those reactions lies on activation energy. So without reaching the level of energy that is required for that chemical reaction to occur, that chemical reaction is not going to occur. Now, the problem is that the cell has to make sure this chemical reaction occur relatively fast so the cell can divide and do whatever it needs to maintain its function and structure. So they rely heavily on enzymes. Most enzymes are protein, which means that they are going to be relatively sensitive to environmental conditions, including temperature and drastic change in pH, which can alter the shape of the enzyme. Yes, I said it, the shape of the enzyme, which means that the 3D structure of the enzyme is essential. Anything that would cause a change in the shape of the enzyme would literally or almost systematically lead to the loss of function of that enzyme. We call this denaturation. We did talk about a particular area of the enzyme that is called the active site. That active site is designed for a specific substrate. Now, there should be a match, almost like pieces of a puzzle. To solve a puzzle, you need to bring together two matching pieces. If the two matching pieces are not matching, therefore you are not going to solve your puzzle. So okay, well, sorry about that. My daughter fell from the swing set. I'm here in my backyard so I can play my video and watch them playing. Anyway, to come back here, we said that the substrate matches the active site, which means that enzymes are going to be very selective. For instance, an enzyme that is designed to break down glucose would not know how to break down a nucleotide such as adenosine triphosphate. Okay, they will not know how to do that. So they are very selective. That's a very important detail to remember. Okay, so enzymes are going to sometimes be relatively complex. Some enzymes actually have multiple active sites. They are modular enzymes. I will not take you into the details of that. In any case, I want you to remember that the cell is very cautious on the level of activity of enzyme. Therefore, the cell is going to find ways to tightly control activity of enzymes. We are going to come into that. But before we get into that, let me come back to the fact that most enzymes are proteins. Some enzymes are non-proteic enzymes, including ribozymes. We did mention that. So the ribosome, that is the organelle in the cell that is responsible for protein synthesis, is going to rely on ribosomal RNA for the synthesis of these proteins indeed. Okay, So the ribozyme, it's one of the enzymes that are non-proteic enzymes. So having said that, the rate of enzyme is tightly controlled in the cell, and all that control is based on the three-dimensional shape. The cell can add something to the enzyme so that the three-dimensional shape becomes either optimal or altered in a way that is going to make that enzyme less active. So I already did mention the pH and the temperature. So there are some optimal conditions that would allow the best activity of the enzyme. For instance, in our body, the temperature would be ideally 37 degrees Celsius for 99.9% of enzyme in our body. The pH would be roughly 7.4 for most of the enzymes. Now, you understand that in the stomach, because the stomach is acidic, well, the optimal temperature, optimum pH that is good for the activity of enzymes in the stomach is going to be an acidic pH. So, because of the active site of the enzyme, we know that we can control activity of enzymes by using competitors. But before I go into that, there is a detail that I failed to mention. I failed to mention the details that some enzymes actually are going to require some addition of factors that are non-proteic factors. Those non-proteic factors can be either organic or inorganic. Now, the inorganic non-proteic factors that can play a role in controlling the rate of activity, either by increasing the activity or by decreasing the activity, those factors are called cofactors. Then we are going to have some cofactors that are non-proteic but organic, such as NAD plus that I mentioned in class, FAD, and NADP plus. Those cofactors that are organic, we call them coenzymes. I want you to remember that detail. Now, some examples of inorganic cofactors are metal ions such as iron, zinc, magnesium, okay, to name only those. Now, to go back to the way the cell can control activity of enzymes, we are going to have what we call regulators. Now, some regulators are going to be activators, some regulators are going to be inhibitors. But let me kind of step back and introduce a new concept, the concept of competition. Now, remember that each enzyme has an active site. That active site can be the location of competition with basically what we call an inhibitor. Now, if a chemical, either the cells design it or the Mother Nature designed it, or we designed it, in as much as that chemical would actively compete for occupying the active site with the substrate, that chemical is doing what we call competitive inhibition. Okay? So we have those inhibitors that are competitive inhibitors. Then we are going to have non-competitive inhibitors, and actually most enzymes are controlled by non-competitive inhibitors. Now let me kind of go back to the structure of the enzyme. Most enzymes are going to have an active site, and beside the active site there is going to be another site that we call an allosteric site. Allosteric. Allo, other, steric, space. Another space on the enzyme on which a substance can come and bind. Now, those enzymes that do have such site beside the active site are called allosteric enzymes. Now here, this is Elik, that's my son. He's 3 years old. Let me show you. Hey, here he is. Okay, he's 3 years old. Okay, well, let me finish this, okay? All right, so let me go back here. So allosteric enzymes are enzymes that are commonly found in the cell, and actually, if I'm not exaggerating, they are the most abundant enzymes in the cell. So allosteric enzymes are going to be controlled by what we call non-competitive inhibition. Basically, in non-competitive inhibition, this is on slide 25 by the way, in non-competitive inhibition, the inhibitor is going to come bind on an allosteric site. That binding of the inhibitor on the allosteric site is going to cause an overall conformational change of the enzyme such that the substrate will not fit perfectly on the active site anymore. So we talked about induced fit. Well, induced fit is not likely to happen when an inhibitor is bound on an allosteric site. So we call this non-competitive inhibition. Now, that non-competitive inhibition is very common in the cell. Now, what we notice is that in non-competitive inhibition in the cell, well, the inhibitor that binds on the allosteric site is oftentimes the end product of a chemical pathway. In other words, let's say that the cell needs an amino acid for protein synthesis, right? To produce a protein. Let's say that the cell needs an amino acid called tryptophan. Now, tryptophan is not synthesized through one single reaction. There are many reactions. So we have reaction 1 that is catalyzed by enzyme